So thank you very much, Heinz-Peter, and thank you to ESOI, the ACR, and the EuCanImage for organizing this session and for inviting me. So I am Karine Seymour. I'm the technical coordinator of the Geminine project, which is one of the brothers of the EuCanImage. We were founded on the same call. I'm also part of the Primage project, and so I will be moderating this second session on data anonymization and curation. So first, we will welcome Michael Rutherford from University of Arkansas, who will explain to us what the problem about data anonymization and curation is. We've already tackled data anonymization quite a bit in this previous session, and he will also share his experience on the Canceri Imaging Archive, which is, I guess, one of the major projects in this area, which we are all looking at in our European projects. And then we will be joined during the panel session by Haridimos Kondylakis, sorry for the pronunciation, from Heraklion in Greece.
And no worries, Karine.
Sorry. Hi, Haris.
Hi.
From the Canceri project, and Martijn Starmans, who will from the EuCanImage project from Rotterdam. And so each of us will give, I guess, so we'll share experience we've had on our respective projects, and then we will take questions from the audience based on those presentations. So thank you very much, and Michael, the floor is yours.
All right, let me go ahead and share my screen here. All right, so good afternoon, everyone, and good morning to my US colleagues. Thank you for having me here today. My name is Michael Rutherford, and I am the lead developer of the POSDA team for the Cancer Imaging Archive, and I'm also a PhD candidate at the University of Arkansas for Medical Sciences. So today I'll be presenting an overview of medical data de-identification and curation. So the ability to replicate and reproduce results is the core element of modern scientific practice, with data sharing really at its heart. So we're all familiar, likely, and it's been mentioned in previous presentations, the FAIR principles, which focus on the concepts of findability, accessibility, interoperability, and reusability. So these concepts are becoming more embraced by the broader scientific community, so it's really made data sharing a much higher priority. So the demand for imaging data and other data objects has exploded in recent years with the popularity of research into artificial intelligence, and the need for these data elements will only increase exponentially as AI matures. So again, as mentioned by previous presentations, there's regulations in place to protect an individual's privacy. There's more than by my count, 120 countries, most notably the GDPR in the EU and HIPAA in the US. Many others have regulations in their countries. And as Davide mentioned, GDPR is so comprehensive. So it really adherence to the GDPR and HIPAA covers most, but some may diverge. So some barriers that we see in the TCIA for data sharing are some institutions and researchers actually consider their data to be intellectual property, and unfortunately they limit or prohibit access to some of these very valuable data sets. So there's also regulatory agencies such as the US FDA that argue for sequestering of data that's used to validate algorithms approved for commercial use. And lastly, and more the kind of the main reason for this talk is patient privacy, data privacy, and informed consent laws and regulations differ between countries, and adherence to these regulations can be difficult. So for clarity, and Magdalena kind of hit on some of this and brought up some of these terms, I like to use the term de-identification as an overarching term for the removal of information which could allow re-identification. So anonymization and pseudonymization are both widely used in the European GDPR. However, I still see them kind of as mechanisms of the overarching concept of de-identification. Anonymization being an irreversible or a complete de-identification, and pseudonymization being the process of replacing these direct identifiers with pseudonyms. So across the bottom here, the GDPR defines personal data as related to an identified or identifiable natural person. So here in the US, we have similar, which is personally identifiable information, or PII, which is any information about an individual. The last term here, protected health information, or PHI, is any individually identifiable health information. So in the realm of de-identification, our main concern is the identifiability of individuals. So whether these patients fall into the role of research subjects or whether they're individuals related to the data set being processed, we have to account for it. So an identifier is a person or a thing that establishes the identity of someone or something. There can be direct identifiers that will uniquely identify individuals, such as names, addresses, and telephone numbers, and also indirect identifiers that can be used to re-identify in combination with other background knowledge, such as sex, date of birth, and geographical locations. So in the US, as mentioned previously, the HIPAA outlines the safe harbor method as a recommended approach for de-identification. This involves the removal of 18 defined PHI identifiers to create a data set that's safe for public use. So this includes names, locations, dates, telephone numbers, numbers for medical devices, email addresses, URLs, IP addresses, national ID numbers, medical record numbers, even veers into biometric identifiers, full face photographs, information about personal or work accounts, certificate and licenses, and then really their last one is an all-encompassing pretty much any other coding or numbers that can uniquely identify a person. So in medical imaging, and I'll kind of focus on this because this is really my specialty. There's multiple modalities that can be used, or image types and modalities. But the dominant file format in clinical practice for image acquisition and distribution is DICOM, which is the Digital Image and Communications in Medicine. So I'm going to focus on this for examples, but these principles that we use for DICOM can really be applicable for any image format or any just documents in general. So DICOM consists of a header and pixel data. The header can contains a data set that's rich with information related to the image. This can include information about the patient, the study, clinicians, institutions, and the machines involved in the creation of these images. So these headers can also contain important secondary research information, such as like physical characteristics like heights and weights, or dosages and administration information related to, say, radiopharmaceuticals. And this information is really critical for certain research, especially in like radiation oncology. So as far as what needs to be de-identified, so the metadata contains both structured and unstructured attributes, which can contain text, numeric, and binary values. The unstructured can be especially difficult to de-identify because it's pretty much just big blobs of text, and you're looking for these. Sometimes it's just a needle in a haystack. So the pixel data can have identifiable information in the content, as well as identifiable features such as faces, secondary indirect identifiers such as, genitalia or unusual or structural characteristics that can be specific to a particular individual, such as the iris or the shape of the skull or even the pelvis. So finally, embedded data can be also burned into the image. So it can be text or as mentioned earlier, maybe even like a serial number on a pacemaker. So most approaches to DICOM de-identification are rule-based. So a pretty good place to start as far as rule-based de-identification is using the DICOM standard de-identification profiles. So the design of these profiles is based on the premise that, with few exceptions, the data elements contain what they are supposed to contain. This isn't obviously always the case, but this has been found to be a pretty reliable assumption, except with respect to like plain text strings such as descriptions, to which human operators have edit access to these and can really just input anything in there. But with my experience, we found PHI in places that you would just never expect it to be. So it really needs some extra kind of human in the loop to make sure that you're able to catch these elements. So the default behavior for the basic profile is to require removal or replacement of all textual descriptive attributes, along with data elements that are known to be direct or indirect identifiers, as well as the removal of all private elements. And private elements are elements that can be inserted by machine manufacturers, they're not so well documented, so sometimes those are a bit of a black box. So additional elements can be removed, replaced, retained, or cleaned based on various named options outlined in the profiles. So as you can see in the top table here are the actual de-identification actions for each of the elements within a DICOM file, and the lower table lists DICOM attributes with columns for each basic profile and the named options with the actions at the intersections. And here on the left, you'll see the red asterisks. Those are actually the options that we use in TCIA. So medical imaging tends to come with accompanying data. This can include clinical reports, annotations, or even documents. So these items require the same attention as the metadata contained within the imaging files. So traditional reports about images, whether they're radiological or pathological, or in the form of clinical notes about images obtained in the course of some clinical study, are usually relatively unstructured and are often dictated as narratives, albeit sometimes they do use templates to constrain the organization of the text. So annotations can include categories, labels, notes, measurements, or similar relatively brief focused bits of information with a specific focus, such as a pixel or a region of interest or a whole image or even a series of images of a patient and or specimen. So lastly, documents can be encoded in DICOM objects or accompany them, which tend to be more difficult to de-identify. It's pretty much big blobs of text and you're that needle in a haystack situation again. So that concludes kind of my overview of de-identification, and I want to go ahead and kind of give a quick overview of the TCIA pipeline and how we handle some of this. So the TCIA submission process starts with the submission of a proposal from clients that want to submit their data. So all these requests are reviewed by a TCIA advisory group, which includes experts from the NCI Cancer Imaging Program, Cancer Diagnosis Program, Center for Cancer Research, and the Frederick National Lab. So once approved, a TCIA submission team supports the submitting site with software and procedures and also guidance on submitting their data. And then the curation process can begin at that point. So the curation process is a standards-based IRB approved process, which complies with privacy, confidentiality, and human subjects regulations. The curation uses the POSDA toolkit to inspect and edit each image for DICOM conformance, linkages to supporting data, the redaction of PHI using multiple iterations of PHI reporting, and human visual reviews. So a key goal for our process here is to strike a balance between preserving privacy while also retaining scientific research value. And a quick overview of the POSDA suite. It's a suite of medical image curation tools developed by the TCIA team. It was originally the Perl open source DICOM archive, but the project has expanded and evolved to include tools written in Perl, Python, JavaScript, and other languages. It's almost become kind of a script repository that you can build scripts in multiple languages and use that within the system in the various architectures and worker nodes. So while much of the functionality is focused on DICOM file types, many functions are now being designed to support other data types, including histopathology. So the suite is hosted in a Docker container, which contains the main POSDA application, the database, and other applications and software tools used in the POSDA curation process. So this architecture includes tools for de-identification, DICOM standards conformance, data integrity checks of the completeness and consistency of RT data sets, referential integrity of inter-object linkages, and checks for spatial registration in multiple frames of reference. So just a little bit about lessons learned from TCIA. Data governance is essential for the quality and value of the imaging and the data being used. And as mentioned, there are many barriers to data sharing. Curation is essential. Computers are good, but to be honest, humans are better, and it's really about finding that balance. While AI is moving forward, it still needs to be trained, and it's got a long way to go in the realm of de-identification. And lastly, clinical data really is more complex than images. As you can see from the presentation, there's a lot of structure to imaging, specifically DICOM objects, and we've learned a lot in the process of building this DICOM standard that other imaging types are kind of taking the cue and using some of the lessons learned. So at that point, thank you everyone, and I will hand it back over to Karine.
Thank you very much, Michael. So now we'll go to the panel session, and I would like first Harris to tell us about Canceri and how the identification and curation is handled within this project.
Yes, in fact, I have prepared just a few slides that we could quickly go through. What is the approach of mine? So the anonymization for Canceri. So this is, as per previous discussion, the Deidentification, fair removal of ambiguous information that could hide the whole workflow. In essence, is this one.
Harris, I think I don't know if it's just me, but you're cutting out. So we didn't hear the last bit. I don't know if anyone else is having the same problem as I.
Yeah, same for me.
Okay.
I am hearing this.
Yes, same for me also.
Is it better now?
Yes.
Okay. So the whole idea here is that we have both clinical data and imaging. So clinical data are being selected and recorded by clinicians. So in essence, they pay attention not to write something that is personal. And then the whole, let's say, anonymization goes to the DICOM images. There we have two anonymization steps. One is the blacklist anonymization, where data providers perform. And then we have a second step that it is transparent to the clinician, which is the whitelist anonymization. So there is a dual stage anonymization, hopefully ensuring that all data that are uploaded are completely anonymized. And regarding the two methods, blacklisting retain most of the DICOM images. And on the other hand, whitelisting retains only a small selected fraction of the DICOM tags. So both of those are being used. Of course, with clinicians, we had to agree which will be those tags and how to preserve them, which to preserve and which were not needed. So in essence, what we do is both blacklisting and whitelisting. And we use the RSNA anonymizer tool to do this.
Thank you, Haris. I myself will share a few slides on the two projects I participate in. The first one is Primage and the other one is Geminine. So I'm just, let me share my screen here. There. So I'm just going to first talk about Primage, which is a project that started a bit earlier than the other ones. It started in 2018, and it's a project on pediatric cancer, on neuroblastomas and DIPG. And here the approach we took in terms of the identifications and data curation. This is the overview of the workflow. We work on pseudonymized data, and we use a tool called EuPid. So EuPid is actually a European solution that was developed in a previous European project, where basically it allows for patients to be included in different clinical trials and different projects. And so each project will have their own pseudonym, but it allows for data to come from different institutions. So that's what has been used for Primage, this EuPid solution. And then the data is stored on a central repository handled by making use of the Quibim precision platform, and it's hosted by the University Polytechnic of Valencia. And to upload this data to the platform, we have several ways. One is to use the Primage web interface, where people can just enter their data, and they use this interface also to de-identify, to pseudonymize the data. Another one is the MedExprime Street solution, which is deployed on premises at hospitals, and that solution can automatically batch extract data from the packs and de-identify the data, pseudonymize it. So there is also this interface with the EuPid solution, and it also makes use of the RSNA CTP. So the RSNA CTP solution is embedded in this solution. And then we have a third way provided by our Austrian colleagues using an AIT solution. And then all the curation process is done centrally on the Primage platform. So these are two examples of some of the curation aspects that we handle. So here I haven't covered the de-identification and the verification of the anonymization process that is already done, but I just want to maybe tackle some other aspects of the curation process. One is the image quality control, and that's in our project is handled by MedExprime, where basically we have developed a system to assess the quality of the images using 6 metrics that assess the signal acquisition quality and artifact detection. And another curation process that we are handling is the labeling of the MR image sequences. As you know, the series descriptions labels are very heterogeneous, and it's very difficult to rely on that to be able to label different sequences. So this work is being carried on by Oula Fay, who is looking at different DICOM tags like series descriptions, protocol names, sequence name tags to try and label the right sequences. And now they're also they are finding that it's not enough, and they're also using other acquisition parameters in the DICOM metadata and using some machine learning classifier to try to relabel those sequences. Now I'm just going to talk quickly about the Geminine project, which was funded under the same call as EuCanImage, and we work closely together. So the Geminine project is receives images and clinical data for 4 different types of cancers from the different hospitals across Europe. The objective is to develop harmonization tools mainly to ensure that data and tools are made there available for AI developers, and we ensure that we have reproducible patterns across images coming from different vendors. And this is the overall architecture. So although it's mainly the same actors as the Primage project with Medexprim, Quibim, and University Polytechnique at Valencia, the approach here is very different, and it kind of looks like what Harris described in ProCanceri, in the sense that the consortium decided to work on anonymized data versus pseudonymized. And I hear all the debates there was in the previous talk about anonymity and anonymized data. And this has been also a big debate in our consortium. And the reason why we went for anonymized data, that was requested mainly by the clinical partners, in the sense that once the data is anonymized, it's no longer considered as personal data, and it falls out of the GDPR regulation, and basically the hospital don't have any responsibility anymore over the data, so they are not considered as data controller anymore, which kind of somehow reassures them. So as per GDPR, we conducted a risk assessment analysis, making sure that we cannot re-identify the data, and I'm taking into account also everything that Magdalena said in the previous talk, that it also depends on the context of use, and so we have very strict organizational measures and security measures over the data to ensure that only allowed people can use the data. But indeed, that means that we also have drawbacks with that, and problems with the data that we are putting in. For instance, a patient cannot opt out from the research because there is no way to know anymore which data is their data on the platform. We cannot also add additional time points on the platform. So that means that all the data sets have to be prepared at the clinical sites, and only full data set with all the time points can then be, and fully curated data can be anonymized and sent to the platform. So that means we also have basically a 2-step de-identification process. We first have a pseudonymization process that is done at the hospital, and we do the data curation on this pseudonymized data. And so what we mean with curation is, and I'm also looking at referring to what Michael just said, is that clinical data is more complicated than images because we have a lot of clinical data in our project. And so basically the curation process will consist in not only checking the de-identification of the images, but also making sure that we have all the time points that we want to have, that the inclusion criteria are well respected, that the consistency of the data, the completeness of the data. And only once we have the data that are complete with all the time points, then we can anonymize the data and send them to the central repository. And with that, I would like to handle the floor to Martijn, who is going to tell us about how this is handled in EuCanImage.
Yes, thank you. So I will very shortly present how we handle this in EuCanImage. So I'm not going to discuss the whole platform, but in EuCanImage, our platform consists of multiple storages. So we have specific storages for the clinical data, the imaging data, and the annotations. And what we're most interested here is indeed the curation and de-identification process. So the TCIA is also in EuCanImage, so we also make use of POSTA, but we also make use of a solution provided by Collective Minds Radiology. This is a company. So this is a tool they provide called Collective Minds Connect, or now it's called Collective Minds Proxy, which is inside the clinical centers. So this tool makes use of a hashing algorithm. So it produces hashes for the safe anonymization of patients. So the pro of using this tool from a company is that it's certified, but the con is that I can't give too many details because they're also not known to me what exactly happens in there. But we also use a combination of blacklisting and whitelisting. And this hash key that is created by this tool is then used in the next step for the clinical data handling. So again, I won't go into too much detail, but we also have the issue here that clinical data is much more difficult, not only because the data is much more heterogeneous, but also how it is stored in the local centers. So some centers still have to gather the data. Some centers have simple databases lying around, and some data centers have, let's say, more advanced storages with proper data models. So for each of these hospitals, we have to see how we can easily get this data out. And in the end, we use the same hash ID that we used in the imaging to, in the end, send everything to the repository. And in this process, of course, we also have to do some curation again. And we try to do this as close to the source as possible to make sure that the curation is proper. But this also, again, depends on the local hospital whether this is possible or not.
Great. Thank you. Thank you very much to the three of you. If there are questions from the audience, please raise your questions in your in the chat or in the query and responses. If you are part of the panelists and you want to ask questions, you can also raise your hand. Maybe I will ask a first question. So we see from our different projects that we have different approaches, even in terms of data curation. We don't necessarily talk about the same things when we discuss about data curation. Some are tackling really just the anonymization, or the de-identification, I should say, making sure that there is no identifiable information in the data. We talk about data completeness and so on, and we also have different approaches about the de-identification itself, whether we do pseudonymization or anonymization. So I'd like to ask Haris about maybe give us his impression, and also the work that's been done among the projects to try and share experiences on that, and what is the conclusion of this work, this common work that we are currently doing on these aspects.
Thank you, Karine, for the question. So as you explained, each project has its own needs and adopts different strategies. And certainly, for example, I only presented the anonymization step of data curation. So even in our pipeline, we have more steps like image annotation, segmentation, co-registration, and the story follows. But I only presented anonymization. And I have to tell you that it is reasonable, depending on the specific choices that each consortium has made based on specific reasons. For example, we will face the problem that if we detect something, we cannot go back to the patient. So, but this is not essential in our case. In other projects, this might be essential. So it depends on the choices that each project does, and all projects, what is common is that we try to avoid bottlenecks, to resolve ethical committees, and all these difficult things that impose huge barriers, let's say. So I would say it's expected, it is common, and I really like to see that we have various approaches and difference, and that we are all able to exchange experiences, discuss what worked where, and to benefit each other.
Okay, thank you, Haris. when we were preparing of this this session, I guess we tried to see what would be interesting questions. But please, if you have any questions, I'm thinking about maybe Magdalena, because it's the topic is, or the speakers of the previous panels who are legal experts on the GDPR and the identification. Please jump in and ask questions if you like. In DICOM, in images in particular, we have specifics about information in DICOM. We've tackled a little bit about private tags, DICOM private tags, and which usually contain very interesting information in terms of research. So the question maybe to you is, how do you handle those private tags, and how can we kind of combine our work and ensure that we have a consistent way to handle private tags? Maybe somebody wants to, maybe Martijn would you like to comment on that?
Yeah, so we continuously have this issue that indeed by default the most strict way of anonymization is just throwing away all the private text, but usually there's a lot of information that's relevant in there. So I guess it's a bit trial or an error most of the time. So you discover at some point that you're missing specific information, then you go back to the original data if it's possible, and you see if you can maintain them. So we have specific ways of looking up specific information in private text. So for example, in MRI, in the fusion weighted MRI, we know for some specific manufacturers where some specific information is stored. But this is just based on trial and error. So I guess most of us face the same issue. So I guess it would be great if indeed we could collaborate on this and we sort of have some best practices in the end, such that not everyone has to go through this process of finding out which private texts contain which information to whitelist. Yeah. I think the TCA already also has some experience in this.
Yeah, I can chime in on this as well. You know, we handle private tags on a case-by-case basis. We are, anytime we run into a new private tag, we search for the documents out there that the manufacturers put out. Sometimes they're easy to find, sometimes they're not. In many cases, we don't even know what this tag is, you know, because it doesn't come up in the DICOM standard with an actual tag name. So it's kind of a search process every time a new one comes up. However, we do document every private tag that we have encountered, and we also add the disposition, essentially how we handled it, whether we kept it, whether we cleaned it, whether we or got rid of it. So we do have what we call the private tag knowledge base. I believe we do have a public file out there, and I'm pretty sure it's out of date because it's really hard to keep up with. And in some cases, we do have duplicates on how a private tag is handled because, and it will have different dispositions because, as you mentioned, a lot of times de-identifications based on the purpose, based on how the data is going to be used. So in some cases, maybe we've removed this tag, but in other cases, if it's pertinent to the research that it's being submitted for, we kept it. So it does take some communication back and forth with the submitters. So our intention is to make this private tag knowledge base public at some point. I've been really pushing for it, and now that I'm heading up the POSTA team, I hope that will come to fruition in the future. However, let me clarify, this will require, if this is going to be worth anything, this will require the manufacturers to have some input to this knowledge base, because without their buy-in, and how I would like to see it is manufacturers have a place where they can submit their private tags to a single repository so that anyone can just look it up from that point.
Yeah, that sounds great. What we now have is we don't per se have this private tag repository, but we mostly use the CTP tool for anonymization. And we do sort of have a set of scripts with different protocols. So I need a strict script, but also based on previous experience, like the brain MRI script. where for some specific sequences, we've made some protocols to handle the most common private text. So they're not perfect, but at least I would say it's a good start for if you start with a new study on a specific use case to have these best practices from previous projects. So I hope within our European projects, we can also do something.
Yeah, so maybe this is a call for a working group, because we're kind of always reinventing the wheel. And so I think requires a collective effort to maintain this list of private tags and scripts. And I like what you said, Michael, too, about, of course, involving the manufacturers of modalities. And maybe I'm wondering if that should, maybe we should, that should be also tackled by the DICOM standard, where basically, ideally, we shouldn't, manufacturers should register basically their private DICOM tags into a database that is managed by the DICOM standards, rather than doing whatever they want. And that would be ideal, which if we could have a reference that is handled by an institution. I guess, and the same goes, I think, with sequence labels, with the study descriptions or any labels that is basically anyone can put anything, and then it's a nightmare to try and harmonize those tags. I don't know if you have any view on that, and past experiences on that.
I believe every one of us have similar experiences. Because things are not standardized, after getting 8,000 of images, we were searching to identify what its image was. It was a diffusion, what it was. It was nothing standardized. Each institute and each vendor had its own, let's say, string, and it was a nightmare. We did it eventually, but you can imagine with a lot of effort. And let me tell you also that I find that vendors, maybe I wouldn't bet on them. However, some, let's say, public institutions, or even, let's say, project collaboration that maintains such a database will be really useful, and then other projects will use it, exploit it, and building a community would help.
I agree, and I think I would have to talk to some of my colleagues, but I believe the standards committee has actually tried to push for this, and I think Harris is right. I mean, the manufacturers don't want to do any more work than they're doing. They're like, we put out a document, and they're done. They just wipe their hands of it and move on to the next things. So, yeah, I agree.
All right. Well, it is 4 p.m. So I think it is time for coffee break, and I don't think we have. I don't know if anybody else has the last question, even from the panelist, Heinz Peter. I don't know if you yourself maybe have some questions or some thoughts about what was just discussed during this session.
Thank you very much for this very interesting discussion. I have a lot of questions. I have more than I could ever ask here, but we need now our bio break. Thank you very much for the speech, for your speeches and the discussion. And we will move on in 15 minutes. Please be back again. Thanks. Bye-bye.
Thank you. Bye-bye.